Hey everyone, and welcome to our video on mitosis. This is part of our Module 2 Part 6 series on cell division, cell diversity and cell differentiation. And in this video we're going to be having a look at specification references 2.1.6c, the main stages of mitosis, 2.1.6d, sections of plant tissue showing the cell cycle and stages of mitosis, and 2.1.6e, the significance of mitosis in life cycles. First thing then, and hopefully a bit of a reminder of what you did back in GCSE, when we're talking about the process of mitosis, we're talking about something that is going to produce two genetically identical daughter cells. And please let's get into the habit of using the correct terminology. Make sure we are saying genetically identical anytime we are talking about the product of mitosis. So don't just say they're identical, genetically identical is the phrasing to make sure is included. In terms of where mitosis is going to be important for our organisms, asexual reproduction, growth and tissue repair. Again, one point of note. Quite often what people do is they shoot themselves in the foot with answers when it comes to the actual uses or the kind of roles that mitosis plays within an organism. Because rather than sort of saying just growth and repair, then people start adding of cells. Please don't, okay? Because it is not the growth of cells, it is the growth of an organism. We are producing a greater number of cells through the process of mitosis, which means your organism gets bigger. Just like it's not repair of a cell, it is tissue repair. So if you've cut your skin, then mitosis is going to be utilised to produce new cells that are then going to repair the tissue. Again, not the cell. So avoid adding of cells to any of these. You can literally go with growth and repair, or if you must, growth of the organism, repair of the organism, or repair of tissues. They would be acceptable. Now, if we actually think about our first example there, asexual reproduction. So what we've got, single-celled organisms like our protoctistans, the amoeba, paramecium for example, they are going to actually replicate through the process of mitosis. And we're also going to find certain plants are going to utilise mitosis to reproduce. So spider plants, which you almost certainly have in one of your science labs at school, and things like strawberry plants as well. The way that they're going to reproduce is obviously asexual. There's no two-partner bit going on here. It's purely one parent plant. And the way that they work is they produce these little runners. So if we've got a cute little spider plant here, then what we end up with is this little runner that comes off the side with a little tiny plantlet on the end. And that little tiny plantlet is an identical copy of that parent plant. It's a clone. So that is asexual reproduction in both our strawberries and our spider plants. We'll also see things like fungi utilising this in our reproduction of things like yeasts. And in animals, we do also see asexual reproduction. Not as often, but there are some examples. So good examples there, sharks and Komodo dragons. Both of those can utilise asexual reproduction, therefore creating genetically identical clones to that parent animal. If we think about our second use of mitosis, the whole growth side, then for a multicellular organism to actually grow, they're going to need more cells. Literally what we've got to do is produce more cells so you get bigger. And obviously what we're going to need here are going to be some cells that are going to be identical to others, hence mitosis. So what we have when we're producing more of these cells, they'll be genetically identical to each other and to the parent cell there. And our third use there of mitosis was in repair, if you remember. So we've already mentioned the whole idea of healing a wound through obviously repairing that tissue. But what we're going to see here in terms of the detail of that, we're going to get these platelets, which hopefully we remember from GCSE, and we've then got something we possibly didn't know at GCSE, the macrophages, that are going to then stimulate that proliferation of endothelial cells and smooth muscle cells. So basically, if you've got a wound, then we are going to have these kind of almost 3 key players, the damaged cells, the macrophages and the platelets, that are all going to have a role in actually causing more of those key cells, the endothelial and smooth muscle cells, to then be produced, therefore repairing that tissue. What we now need to do is delve deeper into the process of mitosis. Now, at GCSE, you probably did not learn the lovely little names of all the little phases. You learned a quick overview of what happened. What we didn't know was actually what happens properly. We didn't break it down into the individual stages and say this stage is called this. At A level, we will. So we've got 4 main stages in mitosis: prophase, metaphase, anaphase and telophase. So you need to know those in the correct sequence. So that's where we have PMAT: prophase, metaphase, anaphase, telophase. If we think about prophase first of all, our first stage in mitosis, then what we will see happening here are those chromosomes are going to shorten and thicken as they condense. And this is occurring because the DNA is supercoiling. So basically we're getting this condensed DNA, which then forms those very noticeable structures in our cell of the chromosomes. Now, if we're talking about our animal cells, we're going to see the centriole dividing, and those 2 daughter centrioles that we've got move to the opposite poles. And remember, the pole is just the end of the cell there. We're then going to have these little threads from a protein called tubulin that forms a spindle between those centrioles. Now, if we're talking about plant cells, those spindles are going to form from the cytoplasm. So animal cells is the centriole, plant cells, they form from the cytoplasm. And the only other thing that's happening at this point is the nuclear envelope is going to break down. The second phase is metaphase. Now, metaphase is where we have our chromosomes lining up along the equator, or we can also describe that as lining up along the metaphase plate. So basically the equator or the metaphase plate is the center of that cell there, and our chromosomes are going to line up along that central point, a metaphase plate, and we're going to have one chromatid on either side of it. We then have our little spindle fibres attaching to the centromere, which is this little region in the middle basically. So what we've now got are chromosomes all lined up along the metaphase plate, attached by their centromere to obviously the spindle. If we have a little look at this diagram in the bottom right, this is a light microscope image. So this is showing us metaphase. Now, obviously in all the little book diagrams we see, all the chromosomes look like lovely little X's, all perfectly lined up. Animal cells aren't quite that organised. What we can still make out though, are that we've got this central point along here, and all of these chromosomes are arranged around that central part. So that's how we can identify metaphase, because we're looking for these structures that are arranged around that midpoint. So make sure you can identify. The third stage then is anaphase. Now in anaphase, we're going to see the splitting of those chromosomes. So what happens is the centromere, which is attached onto those spindle fibres, is then going to be split, and motor proteins then pull those sister chromatids towards the opposite poles. And it does lead with that centromere. So it's kind of like if you've got the centromere, imagine that's the bit that's attached to the motor protein, and the rest of that chromatid is just being dragged along behind it like little tails, and you can see it in the diagram there. The way that this is going to work is we also have the shortening of those spindle fibres, so it's just moving those pairs of chromatids apart so they go to the opposite poles. In terms of our light microscope image, we can see it on the right here. Quite easy to spot anaphase because we're going to have basically these 2 little collections of our chromatids. And you can see they're quite clearly moving to the other sides of the cell to one another. We then enter telophase, and this is where we've got our separated chromosomes reaching the poles. So they're at their end destination. The spindle fibres are going to be broken down because they've done their job. And we then get a new nuclear envelope reforming around each of those two sets of chromosomes. If we have a look at the light microscope image, you can kind of pick out that no longer are you able to identify the individual little chromatids. Instead, it's formed more of a mass. It almost looks kind of like a little blob, I suppose, most of the time. And we've got the two of them, one at each end of that cell. So that gives us our telophase. So when we're talking about mitosis, what we are referring to is the division of the nucleus. So that is literally what mitosis means, the division of the nucleus. So where we've got these 4 stages that we've looked at, all we're looking at is how the nucleus is dividing. So what we've got in those 4 little microscope images beneath are the 4 stages of mitosis, the division of the nucleus. We start off with our prophase, which just looks like a bit of a tangled mess of strings there. We then enter metaphase, where we've got our chromosomes lined up along that equator, the metaphase plate in the middle. Anaphase is where our chromatids are moving to the opposite poles. And then finally our telophase, that's where they are at those opposite ends, and we're then going to get the reforming of the nuclear envelope. So make sure you can identify those 4 stages of mitosis from light microscope images. And it is worth mentioning that one of those things you can be asked to do on an A-level biology paper is a biological drawing. And they have actually asked you to draw one of the cells undergoing a particular part of mitosis previously. So when it comes to biological drawing, you've obviously got the Practical Skills Handbook on the OCR website that I suggest you get a copy of, have a look, make sure you understand all those principles. Because if you do things that are not accepted in biological drawing, you're throwing away easy marks, because they could very well give you something like our metaphase, for example, and ask you then to do a biological drawing of that cell. Now, first thing, please draw what you see. Don't just make it up from the book. So if you start drawing all these lovely little X's perfectly lined up along the actual equator, then you're not going to get marks for that, people, because it's not what we can see. We can look quite clearly and see that's not the same thing. So don't just draw imaginary pictures. We will give you a picture. Please draw what you see. The next one is, as many of you may be doing art, please put your artistic skills to one side. We do not want these little sketchy little lines on anything. Terrible, losing a mark on that one. Remember, we draw the outlines of any particular shape with a nice sharp pencil, and obviously it should be a complete outline. So let's try to make sure we're not rushing it, so don't draw me some kind of weird blobby thing that may or may not join together. Actually take a couple of minutes and draw it relatively carefully. You don't have to get a ruler out and measure everything, etc., but try to sort of eyeball those proportions. And then nice clear sharp pencil lines, no gaps, no overlappy bits, no feathering, no shading. And if you're asked to label it, remember, we're going to have a nice clear ruled line, so take a ruler, no arrowheads with the labels off to the side. Don't start writing across the actual diagram either. But all of this you can find in that Practical Skills Handbook, so get a copy, read it, know it, so that you can obviously get maximum marks on that particular aspect should it come up on your exam paper. The last thing that has to happen is a process called cytokinesis. So basically, once mitosis has taken place, then we have to divide that cell into the 2 cells. Depending on if we're talking animal or plant cells, they do this in a different way. So in animal cells, what happens is that plasma membrane just folds inwards, and then it's going to separate into those 2 individual cells. If we're talking about plant cells, then they need a slightly different process because they've got a cell wall. So we end up forming this thing called an end plate, and that's going to form where the equator was. Now, once we've got that end plate in place, a new plasma membrane and cellulose cell walls form on either side of the end plate. So what we've got is, if we had our original plant cell here, then we're going to have our little end plate forming across the middle. Then we're going to get new cell membrane on each side and new cell wall on each side. And that's obviously going to allow that to split into 2 new cells. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when I next upload a video, and of course head on over to the website where you can find a range of other resources, all in the aim of helping you in your A-level biology studies.